Hello, welcome back. We are continuing on our antimicrobial journey. So in the last mini lesson, we talked about inhibition of cell wall synthesis. Now we are going to discuss all the mechanisms of how we can inhibit protein synthesis. Now, it was a little bit more obvious with cell wall inhibition. The target is being peptidoglycan. We don't have peptidoglycan, but our cells synthesize proteins. We have ribosomes, so how is it that we have these drugs that can inhibit bacterial protein synthesis and ours is not affected? So we got to think of these differences. Can you remember a difference between eukaryotic protein synthesis and prokaryotic protein synthesis? The answer is in the ribosomes. You guys remember 70S versus 80S? So 70S versus 80S, that's right. So there we go. Bacteria and archaea have 70S ribosomes. Now, I keep saying archaea, but there's no archaea that are pathogens, so I'm just going to keep saying bacteria. 70S ribosomes, where eukaryotic cells have 80S ribosomes. So this is the target. These drugs, these antimicrobial drugs, target the 70S ribosome, not the 80S ribosome. So that's that selective toxicity that we've mentioned before that works for these pathogens. All right. So all of them are going to be on one slide, so we're just going to take the time. All right, so the first mechanism of inhibition of protein synthesis is the drug. In this case, it's just an example. You don't need to associate the name with a mechanism, but be able to describe the mechanism. It binds to the 30S subunit. Remember, there's a large and the small. The 50 and the 30S subunit give you the 70S total sedimentation rate. So these particular drugs bind to the 30S subunit, and you'll see it kind of distorts it. It puts it in this little downward curve. And what happens when the mRNA comes to land, the DS subunit comes in, and the tRNAs are coming in to try to find their codon-anticodon match. Because it's put that little dip in there, the mRNA is read improperly. So the tRNAs bring in the wrong amino acids. And so now we've mutated that particular protein and all proteins that are made in the presence of that affected ribosome. So basically the cell is trying to function with mutated proteins and it doesn't work. So it kills the cell. The next one here, tetracycline is an example. It blocks the A site. So remember there's the EPA site, the exit, the peptidoglycan bond-making site, and the arrival site, or the aminoacetyl synthase site. Whew, that was a mouthful. The A site. So these particular drugs come in and they actually land right here. And they're like, nope, arriving tRNA, you cannot land here. So basically what it does is it halts all protein synthesis. So where the first example causes mutations in the protein, translation still continues with mutated protein products. In example B here, protein synthesis is completely halted. It'd be like putting a stop codon in the middle of protein synthesis. The next one, C example, chloramphenicol is a type of this particular drug. It actually comes in and it prevents that peptide bond from being formed. So translation goes on, mRNA comes and lands, ribosome reads it, tRNAs come in, bringing the amino acids, the bacteria is thinking everything's fine, but what's coming out is instead of a linked chain of amino acids that will eventually make a protein, it's just free-floating amino acids. So kind of an interesting way to block protein synthesis. Everything's running along smoothly. They're just blocking that enzymatic activity of the P site. The amino acids are not attached together. This next one, D example here, macrolides is an example, bind to the 50S subunit, right? Our first example was distorting the 30S subunit. This one that binds the 50S subunit blocks the mRNA movement. So it's like putting a clamp right there on the between the large subunit and the mRNA is not allowing it to move through. And so protein synthesis stops. So it's kind of like this one here and this one here end up with the same outcome, is a halting of protein synthesis, but by different mechanisms. One blocks the A site, one binds the 50S subunit, preventing the mRNA from moving through the ribosome, so it just stops. The last two are kind of on the newer versions of protein synthesis inhibiting antibiotics. So the first one is called an antisense nucleic acid. So these are going to be made by those synthetic nucleic acid machines we saw back in chapter 8, kind of those tools of recombinant DNA technology. So if researchers are aware of a particular mRNA strand that codes for a vital protein in a bacteria, maybe an enzyme or something, they can figure out what that sequence is of that mRNA. They can make a complementary sequence of nucleic acid that will, because of the hydrogen bonding, there's that attraction between the A's, U's, C's, and G's if we're talking about mRNA. And the drug is these short pieces of this antisense nucleic acid that will then hydrogen bond to the mRNA. And if you have the mRNA in this antisense, what's the light pink in this picture, you're not going to get translation. Your methionine, first, your start codon tRNA cannot bind to the start codon. It can't even find the start codon. The 30S and the 50S subunit will not come together, and you have an untranslated mRNA, so no protein synthesis for that particular protein. Now, I'm unsure if they focus on just one protein or they can make a cocktail of antisense nucleic acids to target multiple mRNAs. I don't know the details of that particular medicine. And then the last one, I know my head picture's kind of cutting that off. I have my book. It's hard for me to pronounce it anyways. Oxazolidinone binds to the mRNA, blocking the ability of the 50S ribosome to come in. So things are getting started. The initiation complex is trying to come in with the tRNA holding methionine, but then this particular drug will block the ability for the 50S subunit to come in and start the process of translation. So it kind of blocks the initiation step of translation. All right, so that's a lot of different targets for these 70S ribosomes. So we're distorting the 30S subunit, we're blocking the arrival site, we're blocking the peptide bonds, we're blocking the movement of the mRNA, we are using antisense to block even the coming together of the ribosomes, and then we're blocking the initiation complex to start translation. So lots of different targets. Again, you don't need to know the names of the drugs that do these things, but you should be comfortable with being able to describe these various mechanisms. All right, so that is our inhibition of protein synthesis. I think we do cell wall or cell membrane, oh yes, cytoplasmic membrane and metabolic pathway disruption in our next video. Okay, I'll see you then. Bye.